Ladies and gentlemen, welcome back from your coffee break. It's a tremendous honor for me to be chairing this session where Professor Farid Murad is going to be speaking. His incredibly impressive CV is on page 50 of your program book. The only things that perhaps I want to mention is his Lasker Award in 1996, and as often happens when you win the Lasker Award, you normally win the Nobel Prize a few years later, and he did that in 1998. As a young scientist, you often imagine how your path crosses those of great men like Professor Murad. And when I was asked to chair this session, I did that exercise in my mind, although I'm not that young anymore. But I also taught at the University of Texas Health Sciences Center in Houston, where I was a young professor of anatomy many years ago. My organization also recently funded a study to look at the effects of small doses of inhaled nitric oxide in children with severe malaria in Uganda. So there has been a small crossover of our paths in that sense. It's now a great pleasure for me to welcome Professor Farid Murad to give his talk on the discovery of nitric oxide and cyclic GMP in cell signaling and the role of those in drug development. Professor Murad.
Well, thank you very much. I'm pleased to be here. I spoke at this meeting about 6 years ago in Malaysia. So it's been a while since I've been to this meeting, but I'm pleased to be back, invited back. Shortly after the Nobel Prize was announced in October of 1998, my office began to receive lots of phone calls from the high schools, the middle schools, the colleges, universities, churches, and even the mosques, asking if I wouldn't talk to some of the young people about the Nobel Prize and what research I had done. And I did a lot of that for a number of months, but the demands were so great I couldn't keep up with it. So I went to the audiovisual television department in the Texas Medical Center and asked if they would help me prepare a video. And I told them what I had in mind. I wanted to reach out and influence young people to get interested in science and also to learn something about the Nobel Prize. And they agreed to do that. And we worked on it for a few months back and forth and put together on a Saturday morning this video that I'm going to show you. It's very brief, only about 10 minutes. But I think you'll enjoy it, and then I'll give you a lecture about nitric oxide and what it's all about. So let's have the video here. The video is you'll see, in very simple language. I've shown it all over the world. You can find it in several places. It's on YouTube. It's also on the Nobel Foundation website. The Nobel Foundation is really rich in information. It lists all the Nobel laureates since 1901. And alongside of each of the laureates' names is their autobiography, their lecture in Stockholm, and any educational material that they want to add to that. And so it's on that website as well. So if you want to show it to colleagues and your children or grandchildren, it's available. I've shown it to people as young as 4 and 5 years old and as old as 85 years old. And everybody seems to enjoy it, and I think you will as well.
Is, we're going to have to do something about that.
Whoa! What's up with this?
gold medal. And then, of course, there's the money.
Money?
Party?
Royalty?
Yeah. Maybe it'd be better if I show you. May I?
Sure.
Thanks.
Oh, thanks, Fred. Welcome to my world of science and my laboratory. You know, the Nobel Prize wouldn't even be around if it weren't for dynamite. Anyway, Alfred Nobel, the Swedish inventor and businessman who created the Nobel Prizes, was the guy that created dynamite. Nitroglycerin is the explosive chemical in dynamite. And even though it was very dangerous, Mr. Nobel figured out a way to contain nitroglycerin so that it can be put to good use, like to build stuff. You could say his discovery rocked the world. But nitroglycerin has other uses. When Nobel started having heart problems, his doctor actually prescribed nitroglycerin for his heart. But Nobel said, "No way." So he blew it. He figured anything that could blow stuff up can't be good for your heart. Nobody knew why it worked, but it did. 100 years later, Dr. Murad stopped to ask why nitroglycerin helps chest pain, and he shared the 1998 Nobel Prize in Medicine with Dr. Robert Furchgott and Dr. Louis Ignarro for figuring it out.
So you're the dude that figured out why nitroglycerin helped people's hearts?
Well, yeah.
So?
So what? Did experiments. Collected data. Then I found out what I thought was that nitroglycerin releases nitric oxide, and that nitric oxide does a lot of important stuff in the body.
So what is nitric oxide, and what exactly did you figure out about it that got you this Nobel Prize?
Let me show you.
It protects the heart, it stimulates the brain, it kills bacteria, and it's a real gas. It's nitric oxide. No one can say no to NO. Nitric oxide, or NO, is a simple molecule with two atoms, nitrogen and oxygen. And yes, it's a colorless, odorless gas. A scientific sensation sweeps the globe. Nitric oxide is everywhere. This common and toxic pollutant depletes the ozone layer. It's even found in car exhaust and cigarette smoke. But from super menace to superhero, nitric oxide is also found inside the human body and could help send very important messages, which are not from our sponsor. When blood flows through your blood vessels, the inner lining, or endothelium, releases nitric oxide. The nitric oxide signals your blood vessel to relax and widen.
So what?
This in turn lowers blood pressure, the force which the blood exerts on the vessel walls. If your blood vessels make enough nitric oxide to signal your blood vessels to relax, then your blood flows on through no problem. But if blood doesn't flow through, blood clots form. Then
And that's not all.
So, relaxed blood vessels allow more blood to flow, and nitric oxide can have an impact on all different parts of the body. For example, nitric oxide is already saving the lives of babies who were born too early. By breathing in very small doses of this gas, it helps their lungs and improves their breathing.
And that's good.
In nerve cells, nitric oxide can stimulate the brain, affecting things like behavior. Oh, behave, baby. As part of the body's self-defense mechanism, nitric oxide defends against tumor cells and bacteria too. Hi.
It's amazing stuff.
But nitric oxide is no laughing matter, and not to be confused with nitrous oxide, better known as laughing gas. Somebody turn off that gas.
How do you think of all that anyway?
Well, over time, I became very interested in how cells talk to each other. But most other scientists didn't think that was very important.
Dr. Murad figured out that when cells talk to each other, it's like one cell sends an email to another cell somewhere in the body. And the email is the gas nitric oxide. The email can break into another cell and take over how the other cell works. It may contain a message, like instructions for a blood vessel to relax. Or it may contain some other type of instructions. For example, if the message is being sent to a cancer cell, the nitric oxide may kill the cancer and then self-destruct. Hasta la vista, baby. Nitric oxide in your body affects so many things. It's like having a worldwide internet system inside your own body.
So why did you go into science in the first place?
Well, it's really a lot of fun to figure out how stuff like this works. And you don't have to be brilliant to get ahead. You just have to have some goals and be prepared to work very hard.
As a scientist, you get to do something for the first time that nobody else has ever done, and that's exciting. Sometimes your discovery opens the door to a whole new way of thinking, and even more new discoveries. It's really cool. It's kind of like the Science Olympics, and the gold is the Nobel Prize. Teams of scientists around the world compete with each other. It's fun. Who's going to finish first? Who's going to win the prize?
One of you could be a Nobel Prize winner someday. Who knows?
Hey, I had some more questions.
Yeah, me too. Well, I guess we'll have to find out more on our own. Maybe we could check the internet.
Yeah, and look up science and the Nobel Prize.
They're gone. They left some popcorn, didn't they? Yum.
The concept of cell signaling really got started more than 100 years ago. There was a hunter in Michigan, United States, who had an accident while hunting and got shot in the abdomen. And his physician, Beaumont, noticed that this patient would secrete gastric fluid through his fistula that communicated between his stomach and the exterior. And whenever he saw or would smell food, he would enhance his gastric secretion. Well, this information appealed to Pavlov, a Russian physician physiologist in St. Petersburg, Russia, who created dog models with fistulas and pouches to mimic this injury that this hunter had. And he too found that the dogs, when they saw or smell food, would enhance their gastric secretion. Pavlov was very interested in gastrointestinal physiology and realized that the senses and the brain somehow were talking and communicating with the stomach to alter the production of secretion in the stomach. For that work, he got the Nobel Prize in 1904. Today we know, figure out how to advance here. Here we go. Today we know that there are lots and lots of cells in the body that talk to each other. Many, many cells. And this cartoon gives you an example of this process. I've demonstrated here 3 different populations of cells. They can be any population, it doesn't matter. But I'm going to call cell type 1 a neuronal cell that could be in the brain or peripheral nerves. Cell type 2, an endothelial cell lining the inside of the blood vessel. And cell type 3, a smooth muscle cell in the wall of that blood vessel. Cell type 1 wants to talk to cell type 2, and it does this by producing a substance. When it comes from a neuronal cell, we call it a neurotransmitter. But there are other substances that do the same thing when they come from other cells, and we call them hormones, autocoids, paracrine substances, cytokines, growth factors. There are hundreds and hundreds of these messenger molecules. Some are very simple, some are very complicated structures. They're released into the synapse or the bloodstream, and they will then home around the body looking for their target. They identify their target cell by the presence of a protein in the membrane of that target cell that we call a receptor. This first messenger molecule, or ligand, or whatever you want to call it, binds to this receptor in a three-dimensional conformational fit, like a key in a lock. That's where the specificity of the communication takes place. Only the right first messenger or ligand interacts with the right receptor, and only the right receptor interacts with the right ligand. Now, the first messenger doesn't have to go inside of the cell to cause a biologic response. By combining with the receptor, it turns on a biochemical cascade often to increase the accumulation and production of an intracellular second messenger. The first such second messenger discovered in 1957 was cyclic adenosine monophosphate, cyclic AMP, discovered by my mentor, Earl Sutherland, and his colleague, Ted Roll. They were my advisors when I was an MD PhD student in Cleveland. They were looking for the effects of epinephrine and glucagon in liver preparations, trying to figure out how these hormones broke down glycogen to make blood glucose. And in the process, they discovered cyclic AMP. Earl Sutherland got the Nobel Prize for that work in 1971. He had previously trained with Carl Corey and Gerda Corey at the Washington University, where they were looking at regulation of phosphorylase activity to degrade glycogen. So now we have three generations of Nobel laureates, the Coreys, Sutherland, Murad, and also Al Gelman, who was a student with me in Cleveland. And we're all saying that one of our students now has to be the fourth generation, or we're going to be very upset if they destroy the genetic linkage. In the 1960s, about 7 or 8 years after cyclic AMP was discovered, another cyclic nucleotide was discovered in rabbit urine by a couple of chemists. They gave radioactive phosphate to rabbits and found two major organic phosphates in urine, cyclic adenosine monophosphate and cyclic guanosine monophosphate, a cousin of cyclic AMP, slight difference in the structure. And that was the first demonstration that cyclic GMP was a natural product. Well, cyclic AMP went on to be shown to influence the effects of numerous hormones and drugs. The field was becoming quite crowded, very exciting, but I felt as I was finishing my training that I would switch to cyclic guanosine monophosphate, because some of us thought that it too might be a messenger molecule. Well, other messengers have been discovered since: calcium, diisoglycerol, eicosanoids, some peptides. But my favorite is nitric oxide. What is unique about nitric oxide is it's a gas. It's a free radical with an unshared electron in its outer shell. And it's very permeable through lipid bilayers. It can go wherever it wants to go, into the nucleus, into the mitochondria. It can come out of cells and go back in again, unlike other messenger molecules. The other messenger molecules are always charged at physiologic pH. Therefore, to move around, they need carrier proteins and energy, ATP. But nitric oxide can function in the cell in which it's made, or it can come out and regulate the biology of other cells, either nearby or at a distance. So it's a unique messenger. It's a neurotransmitter, it's a hormone, it's an intracellular messenger, an extracellular messenger. There's no other molecule in biology that can take on all of those roles. When it comes out of the endothelial cell, it goes over to the smooth muscle cell, increases the production of another messenger, cyclic guanosine monophosphate, which causes relaxation of the smooth muscle, increases the delivery of oxygen and nutrients to the tissues downstream. Nitric oxide, I think, is a very, very old molecule. I think it was on this planet 3.5 billion years ago, probably, which preceded oxygen, actually. I think it even participated in evolution, but I won't get into that story. But about 50 or 60 years ago, it was discovered that combustion of fossil fuels and other materials that possess nitrogen in their structure, combustion in the presence of oxygen, led to a family of nitrogen oxides. The monooxide NO, NO2, N2O3, etc. These nitrogen oxides were viewed as pollutants or toxic materials that came out of smokestacks, automobile exhaust, cigarette smoke. And the environmentalists got very concerned about these pollutants and toxic materials because as they were released into the atmosphere, they would deplete the ozone layer along with the greenhouse gases to cause global warming. As you remove ozone, you permit ultraviolet light penetration to warm up the earth. But I'm going to tell you, not only is it a pollutant and toxic material, it's a very important material to mediate the effects of lots of important drugs, namely nitroglycerin, nitroprusside, a lot of other substances. And it's a normal substance in the body that mediates a numerous number of effects. The first effects that we discovered were in 1976-77 to cause relaxation of smooth muscle, airway smooth muscle, GI smooth muscle, vascular smooth muscle. We now know it has lots of other effects. It inhibits platelet aggregation and blood clotting. It plays a role in inflammation. It kills cancer cells. It kills bacteria and other pathogens. And the list goes on and on and on. Our first publication was in 1977. Today, 24 years later, 34 years later, there are 130,000 publications in the field of nitric oxide research. It's absolutely incredible. It's become one of the most popular areas in all of medical research and biology. And I can't go through all of the effects. I'll give you some flavor for it as we go on. When cyclic GMP was discovered, some of us began to start looking at how it might be made and how it might have effects in different tissues, if it indeed were a second messenger. And we found that guanosine triphosphate was converted to cyclic GMP, very analogous to the conversion of ATP to cyclic AMP by adenylate cyclase. The enzyme that did this is called guanylyl or guanylate cyclase. There's 7 different isoforms, meaning 7 different gene products make these family of enzymes to make cyclic GMP. The soluble isoform of guanylate cyclase is the receptor for nitric oxide. That's how it works. The other particulate isoforms are receptors for various peptide hormones, the atropeptins, which cause natriuresis and dilation of blood vessels coming from the heart and other tissues, and also E. coli heat-stable enterotoxin, which causes diarrhea, particularly in third world countries, and also a new hormone in the GI tract called guanylin. So these are receptor cyclases. And there's several isoforms where we don't know what the ligand is at the moment. So it's an opportunity to go discover some new hormones for some young trainee. The cyclic nucleotides, cyclic AMP and GMP, are inactivated or degraded by hydrolysis of the phosphodiester bond by a family of enzymes called phosphodiesterases. There are 11 different isoforms, 11 different gene products, very complicated family. Some selectively hydrolyze cyclic AMP, some selectively hydrolyze cyclic GMP, some hydrolyze both. The isoform familiar to all of you, and you don't realize it, is the type 5 phosphodiesterase found predominantly in the wall of blood vessels. And that's how Viagra works. It inhibits that enzyme. And when it does, it permits cyclic GMP levels that are increased by nitric oxide release from the nerves that innervate these blood vessels to go higher and cause greater relaxation. Vessels fill up with blood, and that's the physiologic mechanism of penile erection. Many of the intracellular messengers cyclic AMP, cyclic GMP, calcium diacylglycerol, activate enzymes called protein kinases that transfer the terminal gamma phosphate from ATP to a protein substrate. There are hundreds of these protein kinases, and when these proteins are phosphorylated, their structure, their function, their activity changes. And that's basically how these messenger molecules regulate the biology, is by altering the phosphorylation of various proteins in different compartments of the cell. So the concept of cell communication and signaling is rather simple. A hormone or ligand regulates the production and accumulation of an intracellular messenger, which regulates a kinase, which phosphorylates proteins to cause a biologic effect. The problem is there's so many isoforms, the matrix is rather complicated, and it can take you many years to get it all sorted out to know which protein is talking to which protein. In the process of looking at the regulation of guanylate cyclase to make cyclic GMP, we accidentally found some compounds that would activate the enzyme. Azide, hydroxylamine, sodium nitrite. We put these on cell cultures, slices of brain and liver, and they too elevated cyclic GMP levels. We also put them on smooth muscle preparations. They elevated cyclic GMP, and I thought they would cause contraction, but that didn't happen. I thought cyclic GMP would antagonize the effects of cyclic AMP, which I knew as a student would cause relaxation of smooth muscle. Instead, they caused relaxation. I said, my goodness, the function of cyclic GMP in smooth muscle, airway smooth muscle, GI smooth muscle, vascular smooth muscle, is relaxation. What happens if we add other smooth muscle relaxants to these smooth muscles? What do they do? Well, I used some of these drugs as a resident in the intensive care unit treating patients with heart attacks, nitroglycerin, nitroprusside, to lower blood pressure, to take the workload off the heart after myocardial infarction. So we put nitroglycerin and nitroprusside on these smooth muscles. They, of course, caused relaxation, but they, too, increased cyclic GMP levels and turned out to be activators of guanylate cyclase. And that was a very significant finding, because we now realized that we had a family of compounds for which we coined the term nitrovasodilators that were being converted to a substance that activated the enzyme. These were all precursors or prodrugs being converted to an activator. And it was quite a mystery story for a couple of years to figure out what that intermediate activator was. And some interesting biochemical clues along the way suggested to us that it might be nitric oxide. And we made nitric oxide chemically in the laboratory, put it in our incubations, and sure enough, it activated the enzyme. That experiment was done early December 1996. It was one of those eureka moments where something you thought might work did, and you made a major discovery. This was the first demonstration ever that a free radical, a reactive species with an unshared electron in its outer shell and a gas, would activate an enzyme. It had never been shown before. And it would occur at very, very low concentrations. And we figured out how these nitrovasodilators, namely nitroglycerin and nitroprusside, worked. Now, when you find an exogenous material that works in a system that you're interested in, you should ask yourself, is it mimicking a normal biological pathway? Is there some endogenous substance that's doing the same thing? Opiates led to the discovery of enkephalins. We all knew that when opiates did such and such, there had to be a natural substance doing the same thing. And there have been lots of examples of that in the biochemical and pharmacological literature over the years. So we knew that nitric oxide mediated the effects of nitroglycerin and other nitrovasodilators, and I suspected that it might mediate the effects of hormones by having hormones convert some endogenous precursor to nitric oxide, perhaps by altering the redox pathways in tissues. I couldn't prove this at the time, in 1978, because the concentration of nitric oxide required to do this was extremely low. there were no assays out there for us to measure nitric oxide and its oxidation products, nitrite or nitrate, at nanomolar concentrations. We had to develop new technology, and it took us about 7 years. And then finally, as we did, we proved that this hypothesis was correct. A couple of other laboratories came up with other methods to show this, and we all concluded in the mid-1980s that this prediction in 1978 was correct. And that's what took me to Stockholm, discovering nitric oxide and then predicting it was an endogenous regulator, and it turned out to be the case. It's an intracellular second messenger. At the time, this was all viewed very skeptically by other scientists, our colleagues, our competitors. How in the world could a gas-free radical activate an enzyme, and how in the world could it be an intracellular messenger normally in the body? Well, it all turned out to be the case, and it's a novel sort of pathway for signaling. Shortly after this, Robert Furchgott, a vascular biologist at State University of New York in Brooklyn, had found for the first time that some very important drugs, acetylcholine, bradykinin, histamine, and others, were known to cause lowering of blood pressure, hypotension, in patients and animals, but they always failed to cause relaxation of the blood vessel in the laboratory. And that was because the blood vessel had the endothelium damaged in the process of its preparation. And if he kept the endothelium intact, they caused relaxation. And he discovered endothelial-dependent vasodilators, a very important concept and new finding. And when he treated these blood vessels with intact endothelium, the endothelium released a substance called endothelial-derived relaxin factor, very labile. It had a half-life of only a few seconds, but it went over to the smooth muscle and caused relaxation. I heard him present that work at a seminar in 1980 while I was still at the University of Virginia. And afterwards, I buttonholed him, took him off to my office, and I said, Bob, it's got to work through cyclic AMP or cyclic GMP, and EDRF has to be a reactive species, perhaps a free radical, and maybe a complex of nitric oxide. Let's collaborate and figure this out. And we agreed to collaborate. His wife was with him on that visit. My wife took his wife to Monticello. She slipped and fell, broke her leg. Turned out she had metastatic breast cancer in her leg. And of course, Bob was distraught. He returned to New York to look after her and get her treated. That collaboration never took place. We then moved off to Stanford, and we decided after a while that we would go ahead without Bob's collaboration. And we showed that he was right, that blood vessels release something from the endothelium that cause relaxation, and that something, EDRF, increased cyclic GMP production. And later, we and others discovered that that something, EDRF, was nitric oxide. So we were pretty close when we first started out. So let me put this together for you in a simple cartoon. This represents a blood vessel with the endothelial lining and the underlying smooth muscle. In red are three classes of vasodilators, the relaxed blood vessels, that work by increasing cyclic GMP. Now you can increase other substances and change them to cause relaxation. Increasing cyclic AMP will cause relaxation. Lowering calcium will. So there are other ways to do it. But this is how agents do it through cyclic GMP. The nitrovasodilators are prodrugs or precursors that are converted in the bloodstream or in tissues to nitric oxide. Sometimes it's not enzymatic, sometimes it's enzymatic depending on the structure of the material. The nitric oxide combines with a soluble guanylate cyclase to activate it as much as 200 to 400 fold and at nanomolar concentrations. Very effective substance. The cyclic GMP produced activates a kinase, which phosphorylates a number of proteins, 10 or 12 in smooth muscle. Some of those proteins regulate intracellular calcium levels, and calcium is required for the enzyme myosin light chain kinase, which phosphorylates myosin. Actin filaments, myosin filaments, when the myosin's phosphorylated, they slide together, latch, you have contraction. When they're dephosphorylated, they slide apart, you have relaxation. That's the biochemical mechanism of contraction relaxation and how cyclic AMP, GMP, and calcium regulate that process. Endothelial-dependent vasodilators require the endothelium. Why is that? Well, that's where the receptors are. Their receptors are not on the smooth muscle. Acetylcholine, histamine, bradykinin, and others interact with their receptors only found on the endothelial cells to increase calcium, to activate an enzyme called nitric oxide synthase, or NOS, to make nitric oxide, EDRF, which goes over to the smooth muscle and works the same way as nitrovasodilators, identically. Atriopeptins are a third category of vasodilators. These are hormones initially produced in cardiac atria. The heart is an endocrine gland as well as the pump. And they're also produced in other tissues, and they relax blood vessels, and they increase the excretion of salt and water by the kidneys. They work by activating the particulate isoforms of guanylate cyclase, and the cyclic GMP works the same way as the endothelial dependent or the nitrovasodilators do. There are several forms of the enzyme that make nitric oxide called nitric oxide synthases. These are isoforms again. The genes reside on 3 different chromosomes. There's a lot of similarity, homology between these isoforms. They're very complicated. The NOS2, or inducible NOS, is only found in tissues where there's been an inflammatory insult. Parkinson's, Alzheimer's, myocarditis, colitis, nephritis, arthritis, atherosclerosis. All of those are inflammatory processes. These inflammatory processes release pro-inflammatory cytokines, IL-1, interferon gamma, TNF-alpha, which regulate NF-kappa B to enhance the transcription of messenger RNA for NOS2. So when you see NOS2 transcript or protein, it becomes a biomarker for inflammation. If you don't see it, it means the tissue probably hasn't had any inflammation. These enzymes convert L-arginine, an amino acid in our diet. It's an essential amino acid because our bodies don't make enough of it. You make some, but not enough. It oxidizes the terminal guanidinitrogen to an intermediate hydroxyarginine, further oxidized to form nitric oxide and citrulline. The citrulline can be recycled back to make more arginine. Why do I show this? Well, the enzyme requires a number of complicated cofactors. It's a heme protein. It requires calcium calmodulin, oxygen, NADPH, flavins, and tetrahydrobiopterin. Many of the cofactors are reduced. If you oxidize them, the enzyme becomes ineffective, doesn't make nitric oxide. In fact, it'll make something worse, superoxide anion, which is a trap and scavenger for nitric oxide. And I'll tell you why that's important very shortly. So let me put this together for you now in another flow diagram. Hormones and other first messengers interact often with their receptors. They then modify the intracellular concentrations of these cofactors, which are required for nitric oxide synthase activity to convert L-arginine to nitric oxide. That in turn activates the soluble isoform of guanylate cyclase to make cyclic GMP. That activates a protein kinase. The cyclic GMP can be pulled out of the pathway by hydrolysis by phosphodiesterases. Now, by understanding the biochemical steps in these cascades, we've identified molecular targets. Each one of these steps is a potential site to come in with chemicals to either activate or inhibit that step as potential therapeutic agents for drug discovery. That's how we discover drugs. We do a lot of fundamental biochemistry, genetics, etc., to identify compounds that we want to take in the clinic for important diseases. And we've discovered lots of them. Oops, wrong way. Now, pathways are always a little bit more complicated than you think when you get started. While nitric oxide prefers to increase cyclic GMP production, it can be pulled off in other directions. It can be oxidized to nitrite and nitrate. It can be interactive with other transition metals, manganese, zinc, etc. It can form complexes with thiol groups and proteins. More than 100 proteins get nitrosated or nitrosylated. And a very important reaction is the interaction of the nitric oxide free radical with another free radical, superoxide anion. This interaction is extremely rapid, almost diffusion limited, and when the two molecules are nearby in the same compartment of the cell, they'll make a much more reactive molecule, peroxynitrite, which is very, very toxic. Peroxynitrite will interact with DNA, RNA, proteins, lipids. It does lots of tricks. And invariably, it inhibits the activity of all those enzymes and molecules. One of the things it does is to nitrate the tyrosyl residues in proteins. So when you find nitrotyrosine proteins in tissues, it's also a biomarker of inflammation. In inflammation, I told you, you make pro-inflammatory cytokines. They enhance the production of NO. They also enhance the production of superoxide anion. So you've always heard and read in the magazines and literature that reactive oxygen species and free radicals are bad guys. This is why they're bad guys. They lower nitric oxide and diminish its effective function, and they become toxic. Let's put this together now with some clinical relevance for you. There's a disorder that we call endothelial dysfunction. Your blood vessels do not make enough nitric oxide. This occurs in patients with hypertension, diabetes, atherosclerosis, tobacco use, and probably obesity. Their blood vessels do not make enough nitric oxide, and therefore they're constricted more than they should be, and they compromise the delivery of oxygen and nutrients to their tissues. Why is that? Well, these patients also make a lot of asymmetric dimethylarginine, another metabolite of arginine, that inhibits nitric oxide synthase to make NO. All of these diseases also are associated with reactive oxygen species, hydrogen peroxide, superoxide anion, and they interact with NO to pull it out of the pathway to make peroxynitrite. So we now are beginning to understand why perhaps nutritional supplements with L-arginine, cocktails of antioxidants might be beneficial for some of these diseases. I don't think they're going to cure these diseases, although I've had a few patients tell me that their hypertension's normal, they no longer take their pills anymore, or that their diabetic neuropathy is reversed. I think that's a little unusual. But I think it could benefit the therapies that we're using for these underlying diseases. And I think more clinical studies need to be done with these nutritional supplements to really prove, in fact, that they are working with biochemical markers and tests. And I think those will happen. It just, a lot of the nutritional companies don't want to spend the money, and I think that's unfortunate. So now, where else can this go? I'm going to show you two slides where there are lots of processes and diseases where I think this whole field can benefit drug discovery and therapy in a number of areas. It's not meant to be a complete list. I can't list everything that comes out of 130,000 papers. But I list the things that I sort of follow, some of which we work on in my laboratory. We know that nitric oxide is a neurotransmitter. If you knock out NOS1 in mice, knockout mice, and induce a stroke, their stroke is smaller. If you knock out NOS3 in the endothelial cells, the stroke is bigger. If you do a double knockout, the mice have difficulty with memory. In the water maze test, they can't remember where the float is. So it plays a major role in the brain, but we're not sure how and where. There's a lot more needs to be done there. Pulmonary hypertension is a fun one. Premature babies who weigh 1 or 2 kilos are born shunting blood right to left because in utero, they don't need to perfuse their lungs. They get their oxygen from the mother's placenta, the oxygen. So when they're born, they have a patent foramen and a patent ductus, and the blood goes right to left without going through the lungs, and the lungs have very constricted blood vessels. They have pulmonary hypertension. They're hypoxic blue babies. If you give these babies, and I think one of our speakers later is going to make some comments, who's a neonatologist, if you dilate these blood vessels with low concentrations of inhaled nitric oxide, they improve markedly. They stop shunting as much. They're no longer hypoxic, and it's been a marvelous addition to neonatal therapy in the intensive care units. There are other drugs coming along as well, besides NO. If you give it along with some Viagra, sildenafil, and it works even better. That's a better way to use the drug, I think. What else do we have here? We know it plays a role in wound healing. I think it'll play a role in diabetic ulcers, decubitus ulcers, and diabetic ulcers. It promotes angiogenesis. It inhibits platelet aggregation. Let me tell you a little story here. There's a German thoracic surgeon who I was trying to recruit some years ago to Houston, who was giving low concentrations of nitric oxide and prostacyclin in the extracorporeal pump when he was doing open heart surgery. When you do open heart surgery, the platelets activate, aggregate, and clot in the pump. So the patient's platelet counts drop, and they get more bleeding in the chest. And by giving him nitric oxide in the pump and prostacyclin, you inhibit that process. The platelets get back to the patient, less bleeding in the chest, the OR time is shorter, and the patients recover faster. I am surprised that hasn't become much more popular around the world. We know nitric oxide and cyclic GMP play a role in gene regulation. It regulates many genes, both up and down. It regulates stem cell differentiation and proliferation. By manipulating nitric oxide and cyclic G, we can make embryonic stem cells become heart cells or neuronal cells. That's sort of fun and exciting. So there are lots of possibilities for this whole field. Others have recognized this as well. Most of the major multinational pharmaceutical companies have projects around nitric oxide, one way or another. And there are probably 60 or 70 biotech startups around the world working in various aspects of this, developing new drugs. And I predict that in the next decade, we'll have a number of new agents that'll be very, very exciting for all kinds of problems, I think including cancer. Thank you.
Professor Murad will entertain one or two questions, and then we're going to have our commentator talk for 2 or 3 minutes. So maybe just stay there. Professor George, please.
Professor Murad, have you ever tried to apply theoretical models for the nitric oxide and enzymes or polypeptides that are within your work, such as molecular mechanics? Or
problem hearing you. Have we tried it for what?
to study the interaction of the nitric oxides with the receptors.
On the receptor?
With the, any of the receptors.
The receptor for nitric oxide is soluble guanylate cyclase. It binds with a heme prosthetic group. It binds to ferrous iron in the heme, pulls the heme out of the pocket of the enzyme on the beta subunit. It's a heterodimer. And by pulling the heme away from the pocket a little bit, it activates the enzyme downstream in the catalytic domain 200 to 400 fold. That's the mechanism.
What about the enzymes?
Cancer?
Used for these reactions.
That's a more complicated story,
yes.
As cancers often are. We know that in many cancers, not all, but many cancers don't have soluble guanylate cyclase. But they do have particulate guanylate cyclase. And if you activate the particulate guanylate cyclase, or you introduce a gene with soluble guanylate cyclase activity, you suppress the growth of the cancer. So we've just published a paper with glioblastoma showing that we can inhibit glioblastoma growth in culture and in nude mice implants by manipulating guanylyl cyclase and cyclic G. So is that going to be applicable to other tumors? We're not sure yet. We're looking at that right now. It'll take us another year or two to figure it out.
Please hold the mic close to you.
Hello, can you hear me? You see, I mean, I'm a physicist, I don't know these things, but I will ask a stupid question. The question is that in the whole process, the metabolism plays a very significant part. When things go wrong, so this metabolism rate must be disturbed also. Can you hear me? No?
I can hear you, but I can't understand the dialogue.
Can you hear me now? I don't know how to say that. Say. Can you hear me now?
That's better.
I'm sorry, you see. I'm saying is that in the whole structure that you're describing, I feel that the metabolism is disturbed and the thing doesn't work very well. Is it right? And the second thing is that we know that in the living structures, metabolism rate depends on the mass of the body as a power law. When the system goes down in terms of, does the rate of metabolism also disturb as a function of the mass of the body?
Well, it's very, when you perturb one messenger in a cell, invariably you perturb other messengers because they're all talking to each other. And they all have a variety of functions. Cyclic G affects cyclic A, it affects calcium, it affects lots of other things. We also know that nitric oxide regulates the mitochondria to make ATP with the energetics of the mitochondria. Is that because NO combines with some of the cytochromes and electron transport? Perhaps, we don't know. It's an area of interest. So yes, it becomes very complicated looking at all aspects of metabolism. And it's always very, very difficult to know what is primary, what is secondary, what is tertiary. And what we do is we create cell culture models because we'll never answer those questions in an attacked animal or a human. You can't. So we go to simple models that lack one of the enzymes in the pathway. Either they have suppressed messenger RNA, or they have deficiencies or mutants or whatever, and that permits us now to test our models and concept, and then we can take it to the next step to a more complicated model.
Can I make a comment, please?
A very, very quick comment there, because we've really run out of time.
Hello. I just want to make, as a theoretical physicist, I've been taking interest for the past 20 years, and I've been close in touch with Professor Ubaid Siddiqui from India, Professor Louis Johnson from Oxford, and Professor Kenichiro Miura from Japan. So, and Professor Abdul Salam also took great interest in this matter. The thing that I want to pass on, you see, the chromosomes. The second smallest chromosome was unraveled in 1999. In fact, the 2nd of December in Nature. And the 6th of December, there was a seminar in Cambridge at which I was present. Now, the three things that I'd like to pass on to young people who are interested, and not so young people, sort of thing. Now, the sequencing, as you know, the second smallest had 33.4 million monomers. And altogether, now they have all been unraveled, 23 of them, the 3 billion monomers. Now, many people thought that the sequencing would give insight to various things. Now, it has turned out, we've discussed this matter in detail, that you can't get any kind of insight on anything except very, very specific matters. So that remains a problem. What does the sequence mean? And then there are other two matters. As you know, Professor Salam took great interest in this matter at the end of her life, that all the biological molecules are left-handed. Biological man. That's the main great mystery. And he and later on Professor Louis Johnson also tried to use this to throw light on parity violation or connect the two. And this was never possible. And finally, Professor Kenichiro Miura, whom I met in Japan, Professor Salam used to say that the 20th century has been the physicist century, and the 21st century will be the biologist century. When I told this to Professor Kenichiro Miura, you know, he's a distinguished biologist at Gakushin University in Japan, he said, yes, that's true. But in the biology of the 21st century, the physicists will have to play an important part. But I'll leave it at that for the moment.
I think in the 21st century, all sciences are going to become important, and they all communicate with each other, and every discovery is going to influence another discovery down the road.
And nitric oxide will be the molecule of the century. Professor Murad, if you'll allow me to, on behalf of the Academy, to present you with the shield of the Academy and my latest book, The Grandest Challenge. Thank you very much. Thank you. We now call upon Professor Dillman to give us a commentary on the talk.
Yeah, thank you very much, Professor Murad. Now, being a clinician and as a neonatologist, we are using nitric oxide very widely in our clinics. Due to pulmonary hypertension of the babies. Pulmonary hypertension means, because there are some people around here out of medicine, is the problem with the pulmonary arteries, especially the term babies, sometimes after mecoim aspiration. We are using nitric oxide very widely in those cases, and the results are very nice. Before nitric oxide, we were sending the babies to ECMO. ECMO is a very difficult procedure for the babies. You have putting two cannula to the arteries of the babies, and then we were giving them to extracorporeal membrane oxygenation. It was a difficult task. After nitric oxide, now we are giving nitric oxide through the ventilatory support. It's an easy way, so we are very happy with nitric oxide and the results. While we are speaking about the nitric oxide to the students, we are summarizing in one sentence through L-arginine with nitric oxide synthase, you can produce nitric oxide. But it's not so easy, you know. Here, from 1957, Professor Murad is dealing with the same subject, more than 50 years. And then this morning, I screened the PubMed. He has 146 articles in the same subject. The last one will be published at the end of this month. It's online. It's a big task and it's a big effort. I know he's a good sample for the scientists, so I would like to thank him for his efforts on behalf of our patients. I hope you are going to find new areas for the application of nitric oxide in the near future. Thank you very much for your effort.
Let me make one comment. I've always had a rather large laboratory. I just moved about 6 months ago to George Washington University, and I'm going to decrease my laboratory from about 20 or 24 people down to about 8 or 10 people. So I'm now selecting a couple projects that I really want to work on hard. And we've always worked on things that have a very high risk that haven't been done before. That's where I get excited, is if we could approach a clinically relevant problem in the laboratory and make a major contribution, that's what excites me. And we picked cancer, stem cells, and bacterial diarrhea. all of which are very substantial diseases. And I think we have some interesting leads already in the last couple of years, so that'd be fun to get it resolved.
Maybe also do malaria.
I'll leave that to Peter Agre.
Thank you very much, Professor Murad, for a very stimulating talk and a summary of incredible work over 50 years. Thank you very much for gracing us with your presence, and we look forward to seeing you again at the next meeting. Thank you.
Thank you, everybody. Thank you, Professor Murad. Thank you, Professor Dar, Professor Dillman. We're running late, as you know, so we'll immediately, after 5 minutes, resume the 2 sessions in Salwa 2 and Salwa 3, the 2 parallel sessions. So kindly make your way to the 2 sessions right now. Thank you.